So, good morning. Welcome to the plenary event, Search for Life on Mars, Lessons from Spain. The plenary will be moderated by John Rummel from NASA headquarters and myself. I'm Gerda Horneck from the German Aerospace Center in DLR. So there are certain questions which have intrigued mankind since long time, and these are we alone in the universe? Is the Earth the only planet that harbors life? These questions are, and the solutions have searched mainly in the realm of mythology, philosophy, and maybe also science fiction. But with the advent of spaceflight, we are now in the position to approach these questions experimentally in a scientific way. We can send spacecrafts to other planets of our solar system and search in situ there for life. But what planets of our solar system are habitable? That's the first question we have to solve. Which planet has really the conditions to support a life? And there we are in a dilemma, because currently the only life form we know is life on Earth. We do not know of any other planet that carries life. So in order to define the requirements of life, we have to look at our biosphere and to see what are the limits of life and what are also how wide we can stretch the conditions of life in order to define the requirements of life. And if we do this, one of the planets is of very high importance and a good candidate to look for, and we are lucky that's not so far away from us. That's our neighbor planet, Mars. And so we will, during this plenary, try and tackle the question, why is Mars a habitable planet? And that is a requirement to support life. Maybe not now, maybe in the past, so through the whole history. And we will also link the different exploration programs which have been put forward by the different agencies to investigations that are required here on Earth with life under extreme environments. And we especially look at the research that's done here in Spain on the Rio Tinto. And with this, we want to start the different talks, and after that, you will have the opportunity to discuss it with the experts. The first expert who will give a talk is Dr. Frances Westall. She is a Directeur de Recherche at the CNRS in Orléans, in France. And she has especially contributed to our knowledge of the early biosphere. She's a geologist and bacterial paleontologist or paleobiologist, and she has especially brought us some insights in the very early forms of life as indicated by fossil research. And she will give us a talk on setting the scene for search for life on Mars. Please, Frances.
Thank you very much, Gerda. I was very pleased to receive this invitation to give you a talk on setting the scene.
Can't hear you. No.
Yeah. Okay, that's better. Well, actually enjoyed myself having carte blanche to prepare a nice talk on setting the scene. Is it okay now?
She says it is. We'll do a quick test.
Yes.
Yes, thank you. Okay, you can do your own stuff.
As I was saying, I enjoyed myself preparing a talk on setting the scene and just letting my imagination go wild. Unfortunately, Gerda this morning is very severe, and she said, "You have 5 minutes, Francis," so I had to cut out half my talk immediately. Anyway, so setting the scene for setting the scene for the search for life on Mars basically means finding out about the habitability of the planet, the context of life on the planet. As Gerda said, I'm a geologist, so although I'm interested in the early biosphere on Earth, first and foremost comes the geology, the context, the environmental context for life. And looking at the context on Mars, we need to think in terms of different time periods, different scenarios, because there's Mars, early Mars, there's Mars today, and there was Mars in between. So what we're looking at is the habitability of Mars through time, and especially for particular periods. Is this a pointer? No. Is this a pointer? No. Okay. No, thank you. Especially the beginning, Mars, and the origin of life. Could life have arisen on Mars? And once it was there, or if it came in from elsewhere, could Mars have supported life? Could Mars have supported life on a long term, in terms of billions of years? Could there be life on Mars today? So I'll quickly start off with the first part, which is the nitty-gritty bit, Mars and the origin of life. What do we need for life? We need a number of ingredients. We need liquid water, an energy source, a carbon source, and nutrients, and put it all together in some kind of primordial soup, and just hope that something comes out. Were these on Mars? Well, the energy source, we've got abundant sunlight. Thank you. We have abundant sunlight. There's geothermal energy from hot springs and underwater vents. Also, geochemical energy from redox reactions at the surfaces of minerals and volcanic particles. This is volcanic sand. So there's abundant source of energy, different kinds of energy on Mars, early Mars. Now we need carbon. Where can we get carbon from? Again, abundant source of carbon. All the planetesimals contained carbonaceous material in them, kerogens. Carbonaceous material came in with the carbonaceous meteorites, micrometeorites. There's also carbon in the comets, and also in the atmosphere, and CO2 in any water that was on the surface of the planet. Now, life also needs nutrients. Amongst some of these are nitrogen, phosphorus, and sulfur, amongst many others. All these nutrients were available on the surface of Mars. They're available from the minerals, the fluids on the surface, and the gases in the environment. So basically, there were all the ingredients for a primitive cell to arise, and if, for one reason or another, life did not appear independently on Mars, there's a possibility of it hitchhiking on a meteorite that came in from another planet, such as Earth or even Venus. So there's no reason to think that life could not have existed, either arisen or existed in the early history of the planet. Now, we were interested in the physical habitats for life as well. We have the ingredients, we have the nutrients, the stuff that life needs to survive. But what about the habitats? Now, in thinking the habitats, you don't really need to think in terms of a planet-wide or global scale habitats, for instance, continents and oceans. The fact that probably early Mars did not have as much water as we thought it did. It was not covered by an ocean as was the early Earth. that's not important, because if you think in terms of on a microbial scale, the microbes are small. They can live in very small habitats of the order of tens to hundreds of microns in size. And even these habitats can be very ephemeral. So they don't really need a lot of space and a lot of time, but they do need a constant support of nutrients and water and carbon. So because in the small environment, they will quickly use up the nutrients unless they're replenished on a regular basis. On the Earth, this kind of replenishment on the long term is undertaken by plate tectonic activity over periods of hundreds of and thousands of millions of years. This did not happen on Mars. So what about their survival strategies? They could survive in an environment which is potentially hostile by using various strategies. For instance, microbes can produce spores, or they can go into mummified form and just sit and wait until the necessary water and nutrients and what have you come along again. How long? For how long? We don't really know how long life can just survive in a kind of dormant or mummified or spore form. There have been experiments or analyses of permafrost in Siberia, which by our Russian colleagues, who found cells dating perhaps back to 2 or 3 million years. And these are mummified forms. But billions of years, this is rather a long shot. Just going back to this question of scale again, these are some images from the early Earth. These are fossilized microorganisms that come from rocks 3.5 billion years old on Earth. And it's just to give you an idea of the scale, the size of the organisms. The scale bar here is 0.5 micron in size. These structures are 0.5 micron to less than a micron in diameter. So they're really small. And the microenvironment that they need is pretty small. But there are also some larger structures, such as this stromatolite here, again, from rocks that are 3.5 billion years old. Such a structure would be readily visible at the surface of Mars if it ever existed, if something similar existed there, because you can just see, you can see it with the eye, with your own eyes and with camera and so on. But the small structures are not very readily visible. So early Mars would have provided quite a variety of actual physical habitats. This is a painting actually of the early Earth, but we can transpose it to Mars. You can see fumaroles, hot springs, hot spots, water trapped in impact basins or even volcanic caldera, atmosphere rich in CO2, very little oxygen. So this image gives you an impression of what the early environments of the planets such as Mars and Earth could have been like. What about Mars and life today? Well, if early Mars had a certain amount of water, perhaps in impact basins, it certainly doesn't today. Mars is a very dry planet. So on the present-day surface of Mars, because of the dryness and oxidizing conditions, the UV radiation, the surface is extremely inhospitable. But we know that life can survive in very extreme environments on the Earth, and life could possibly survive in a subsurface aquifer if there is a supply of nutrients and liquid water, of course. So I'm finishing here, otherwise Gerda will crack the whip. And with the idea that if there ever was life on Mars, we're more likely to find traces of ancient life rather than modern life, just because of the difficult surface conditions. And if we're looking for traces of ancient life, there's half a planet full of rocks. All the southern highlands of Mars have rocks old enough to contain traces of this ancient life, if it was there. So I conclude that the traces of life are going to be extremely subtle if we're looking for them. But firstly, because the primitive life was very, very small. It leaves a very small amount of organic carbon if you're wanting to do chemical measurements. And what we basically need to do is to work towards sample return missions to bring back suitable rocks. And in the future, long-term future, because there's nothing better than a geologist in the field, send geologists to Mars. Thank you very much.
Thank you very much, Francis. You have really set up the scene, but please keep your questions until you have heard the whole story, and we continue and we hand over to John.
Well, we're lucky today to have a couple of space agency representatives with us. Michael Meyer from NASA is one of them. Seen here in this picture taken in beautiful Barcelona in 1993, where we were lucky as well to have the first Mars lander program scientist, Dick Young, available, along with a couple of other guys and Mike. And Mike is a natural inheritor of the Mars lead program scientist title at NASA headquarters. I'm told he has a university degree and a PhD from Texas A&M. And Mike will be talking to you today about future chances to explore Mars that may lead to this. That's not Dick Young in the upper picture, but that could be Mike Meyer later on in the lower picture. And with no further ado, let me introduce Mike.
Well, thank you, John, for that most interesting introduction. What I would like to do is do a quick overview of what we have planned for exploring Mars with NASA programs, but then also segue into the things that we need to do here and a little bit of philosophy on why I think analogs are a particularly important area of research that can be done here so that we can much more effectively explore the red planet, and in particular for the search for life. So with the first slide, in case you didn't hear my name, in this decade, we have quite a series of missions going to Mars. In fact, it really gives me great pause to think about how lucky we've been in sending a spacecraft to Red Planet and how we've learned so much. Right now, we have operating 6 spacecraft: Mars Global Surveyor, Mars Odyssey, Mars Express is ESA's mission, and Mars Reconnaissance Orbiter, which is, there'll be a press conference at the end of this week, tomorrow, showing some of the latest pictures. And then also 2 rovers on the surface of Mars. And we found a lot. One of the things we've been doing, following the mantra, the theme of let's look for the water, because that will be a key to understanding whether or not Mars has ever been a potential habitat for life. Well, we found evidence for water and now have some understanding of a particular environment where it existed, and certainly want to explore more to find out what other types of environments may have also had liquid water standing on the surface. One of the things to point out is that in 2007, next year, we'll launch a lander to go to the polar regions and dig into the surface soil and ice to perhaps get a climatic history over who knows how many years, and in fact see if any organics might have been preserved in the subsurface in that ice. Also, the Mars Science Laboratory, which will be launched in 2009, will have a very capable analytical suite inside, be able to do definitive mineralogy with XRD, XRF, that's X-ray diffraction, X-ray fluorescence, and also do quite a range of molecular weights of organic compounds. So this rover will be able to cruise over the surface, drill into rocks, and do samples, and probably stands as our best chance to date of getting a good idea of whether or not we've ever had a habitable area on the planet Mars. What that does is it sets the next decade for what we hope to do on the red planet. We're in the process of having a competition for PI-led missions right now. We have proposals in hand for doing scouts in 2011, and that's pretty much the nature of the mission is solely dependent upon the quality of the proposals and whether or not they'll actually propose something that will work. 2013, we hope to follow up with another orbiter that will now address things such as aeronomy and get an idea of the evolution of the atmosphere of the planet Mars, so we have a better idea of when Mars might have been habitable and for how long. Coming into that, we reach a kind of a period of indecision. Basically, we want to understand what we're learning today to help affect what missions will be in the future. So in 2016 and 2018, those two opportunities, we hope to do 2 out of the 4 missions shown there, such as another scout competition, a network mission to get at the geology, geophysics of the area, understand the evolution of the planet itself. Perhaps we want to expand our geological repertoire by sending 2 more landers of the same scale as the Mars exploration rovers on the planet now. Or we will go for an astrobiology field laboratory. And basically the idea is to follow up what we think we've learned in terms of Mars being a habitable planet and actually go and measure a particular area, hoping to find evidence of either prebiotic chemistry or maybe even remnants of life itself. And then after that, I'm sure we'll have the opportunity to follow up with the Mars sample return mission as sort of the be-all and end-all of answering many of the questions that are left hanging from Institute research. One of the things I'd like to point out is that it's important to have a strategy of what you want to find out and how you should go about doing it. And in 1995, we published the ExoBiology strategy for Mars exploration. And basically, there were 6 very basic recommendations that came out of that, some fairly specific and others much more general. And one of the things that amazes me, particularly having been in the government more for over 5 years now, is that even with the strategy, that we've actually been able to address many of the things that we've recommended. For instance, we did fly a gamma ray spectrometer on the Mars Odyssey in 2001, getting the distribution of elements across the surface. We've flown a mid-IR spectrometer also in Odyssey to give us an idea of the distribution of mineral deposits. And the Mars Reconnaissance Orbiter and also the Mars Express have near IR to also give us greater definition of mineral distributions on the surface. We've developed a mineral identification capability on landed missions in terms of looking at XRD, looking at Mossbauer and also with APXS with the MER rovers. And certainly when we get Phoenix and also the Mars Science Laboratory on the surface, that will greatly improve that capability. In the lighter blue or teal colors, we have things that we haven't done quite yet, but we'll be doing in the near future with missions that are planned, such as acquiring samples from protected areas, able to get into the subsurface of rocks and soils, and then also continuing our basic research and the intellectual underpinnings of understanding how life is possible in extreme environments and what we should do to go about looking for it. And certainly the last one that we'll be doing in Mars 2009 is looking at detailed information about the volatile phases and also particularly doing analyses of hopefully organic compounds that we might find on Mars. But now let me go into sort of get into the intellectual underpinnings and actually mission operations, how we go about exploring on another planet for something that we think we know and we certainly only know in one case, and that is life on Earth. This is a very good example of how you might be fooled, particularly if you're doing a robotic mission. This is a picture from the Atacama Desert, and that pen is pointing to a crustose lichen. If you didn't know any better, you just think that was a salt deposit, but in fact this is a complex organism that is a commensalism between a lichen and it is a lichen between a fungus and a plant. Of course, if you found a pen on Mars, that'd be pretty easy to tell you that there's life, but the harder part is understanding of recognizing that a crustose lichen is in fact something alive. So the NASA has started a program in astrobiology called astrobiology, science, and technology for exploring planets, and the specific purpose is to go about we require robust instrumentation on mobile platforms, and it'll do efficient operations with a high degree of autonomy on another planet. So for this, we needed to come up with a scientifically driven exploration way to do things in extreme environments here that are analogous to the habitats that we think we'll find on another planet, such as Mars. And so analog research basically expands our known limits to life on Earth and develops methodologies for detecting past and present life and advanced methods for exploring rugged, novel environments while meeting astrobiology objectives. Interestingly enough, the picture from Victoria Crater that was taken just this past week doesn't like to show up in this platform, but I'm contrasting it to Rio Tinto, which happens to be a very good analog for Mars. And what I'd like to do is just touch on two of the things, research areas that we've been sponsoring in Rio Tinto to demonstrate how analog environments can help us very much in understanding some other planet. And then I'm sure that Ricardo will talk some more about that. So the Marte project, basically the idea is you go to Rio Tinto. This is a acid drainage area, which was not only famous for the minerals that have been taken out for millennia, but also more recently, the rich life that's been found in the waters themselves. And so one thing is to drill down through the sides of this river and look at rocks and understand what the rocks will tell us that indicate that there's actually life in the Rio Tinto itself. Another approach is, in fact, looking at Rio Tinto in mineralogically, and Omega has found deposits of kaiserite gypsum and polyhydrated sulfates, and the idea is that is there something here on Earth that gives us an idea of what those minerals really are and what they may look like. And so here, a sample from Annabelle's garden of the Rio Tinto matches up with jarosite, which is found also by MER Opportunity, and demonstrates that Rio Tinto is in fact a very good habitat for showing sulfate mineralization from acidic waters, which so now we have a nice parallel between here in Spain and the surface of Mars. So Rio Tinto is a spectacularly beautiful place, and it's well worth going to just for that, but also it may be the only way for some of us here to get close to Mars. So with that, I'll end and permit the next speaker.
So with this, we are already in Europe, and as you might know, also the European Space Agency has started a program to explore Mars. We have the ExoMars, we have the Mars Express mission, the orbiter, and we have now in the exploration mission a flagship, the first flagship, and that is the ExoMars mission. And who can give us better insight into that than the ExoMars project scientists of the European Space Agency, Jorge Vago.
Thanks, Gerda. Good morning. I'll try to go through my talk in the 5 minutes that I'm supposed to be able to cover it with. And so I'm going to start by telling you the punchline. The punchline is how important it is, if we're going to address the search for life, to have two things on the surface of Mars: mobility and access to the subsurface. That's point one. And point two, if we're going to search for organics, we should try to couple the search for organics with an understanding of the oxidants and their effect on the organic molecules on the Martian surface. Having said that, I'll move on. I will concentrate on ExoMars because it's maybe the most visible part of what we do in ESA in order to prepare for future Mars exploration. ExoMars was recently approved, was approved last year at the 2005 Ministerial Conference in Berlin, and it is a search for life mission. It is, its main objective is to search for traces of past and present life on Mars. But we also want to characterize the geochemical environment in the subsurface from 0 down to 2 meters in depth. And we want to understand if there are any hazards that we have to consider in our preparation for future human missions. Oops. We also have technological objectives. In Europe, we still have to demonstrate that we can land large payloads on Mars, and we also need to build up our expertise in terms of mobility and the drill to access the subsurface. And lastly, we see this mission as a stepping stone towards Mars sample return. So, what does ExoMars look like? It's basically a rover powered by solar panels that will be able to cover a distance in the order of a few kilometers, much like the Meyer rovers of NASA are doing now. It will contain a very powerful analytical laboratory to search for biomarkers. And it will also be able to characterize the geological environment in the sites that the rover will visit. Here I show you the areas of Mars that we can target for landing. And we have a maximum altitude of 0 meters with respect to the molar reference level. So basically we can go to areas there that are yellow, green or blue. One of the major assets that we have on this mission is mobility. Here you see an artistic rendition of a rover coming out of the ramp. And I'll go through a few points that we had to try to address in order to put the mission together. The first one is where to search, and we have heard already that life needs liquid water. And if we look at the extant life, present life on Mars today, the environment is very harsh. The UV radiation will kill or degrade any organic molecules exposed on the surface unprotected. We have oxidants that were hypothesized after the Viking mission that will also eat at organic molecules in a distance, depending on which model you consider, in the order of centimeters to maybe a meter or more. And we also have ionizing radiation. So all of these things point towards looking for life on protected sites, that is, within the geological record. Also, in the case of extinct life, if we are looking at the remnants, the molecular remnants of past life, maybe 3 billion years ago, we also have to look for places in which these molecules might be well preserved. And here again, the focus is on looking within appropriate rocks and deposits, and also in the subsurface. This is, I won't dwell very much on this, but just to point that this is a simulation of what is the survivability of organic molecules, in this case amino acids, to ionizing radiation, after several billion years of simulated ionizing radiation influx. And what this points to is that if you want to be able to target organic molecules that were there at the beginning of the history of the planet, you have to be able to go down to depths in the order of 1.5 meters to 2 meters at least, preferably even more, but that's what we can do with a mission. So that is why a major element of ExoMars is the access to the subsurface, which we accomplish by means of a drill. In terms of the international context, you have heard about the Mars Science Laboratory going to Mars in 2009. This will be an extremely able mission. It will be able to, it's powered by radioactive thermal generators, and it will be able to cover distances in the order of hundreds of kilometers, maybe 1,000 kilometers. So for the first time, we will be able to cover large areas of Mars and possibly more than one geological conformation. And we feel that ExoMars fits very well after MSL by providing this access to this third dimension, which is depth. So, we feel ExoMars is a very exciting and timely mission for Europe, and that fits very well within the international concept and context of missions. One of the things that for us is key is Viking in 1976 did not find any organic molecules on the surface. So why was that? There was this hypothesis that there were oxidants that were destroying the organic molecules. We feel after 30 years after Viking, it is high time that somebody goes there and measure these organics and characterizes them. We know that Mars gets in the order of 250 tons of organic material delivered every day by cometary bombardment and meteoroid infall. Where are these organic molecules? They should be on the surface. Well, on the surface, they would be destroyed maybe by UV. But when we go beyond this oxidant horizon, we should be able to see them. So this is something that for us is very important. Match the presence of organic molecules with the presence or absence of oxidants, and be able to map this interplay on the top 2 meters. And finally, we also expect to be able to address important issues of environment that will allow us to prepare for future human missions. And I will stop with this. Thank you very much.
Well, it gives me great pleasure to introduce Professor Ricardo Amils, who has come down from Madrid, where he is a professor of microbial biology, a professor of microbiology, and focusing on what microbes do for a living. Effectively, Professor Amils thinks about microbes from their perspective and also the perspective of how we can use them. He's a senior scientist also with the Spanish Centro de Astrobiologia, which is supported by the Spanish government. The Center for Astrobiology is an innovative partnership looking at how life can exist elsewhere in the universe. He's educated in chemistry, PhD from the Autonomous University of Barcelona, and spent some time in the United States before coming back to serve his country and his students. His interest includes evolution and reconstruction of the foundries of the cells, the ribosomes, biomining, bioremediation, microbial ecology of extreme environments, and strangely enough, astrobiology. I'll also say he's a very good host. So without further ado, I'd like to introduce Professor Amils.
Well, thank you, John, for this nice introduction that probably I don't deserve. But anyway, today I would like to talk a little bit about Rio Tinto. I mean, you heard too much about it, so now let's see the action. So I think what we learned in the recent years from many different types of missions is that there is no doubt that in Mars exist sedimentary rocks probably generated in acidic conditions. So what we can do before, these guys design all these beautiful missions to go there and take samples. Well, and I think in the meantime we have to get ready, and one possibility is to study environments on Earth that can tell us a little bit about what kind of instruments mainly we have to design in order to detect signs of life if they exist. I mean, Rio Tinto is one of the two possible models. I mean, because in Spain we don't have enough money in order to get an Alvin to go under the water to study hydrothermal vents. But we have a beautiful river that is not far away from here, at 100 kilometers. So if you have an option, I recommend it to visit. And I don't think I have to explain that the name of the river claims the color of the river. You know, it's called Rio Tinto because it's Tinto. I mean, I suppose that after the paella last night, the people that didn't know what Tinto means, it knows. Now it knows. Rio Tinto is 100 kilometers acidic river. The main pH is 2.3 due to the buffer abilities that ferric iron has, and it rises in the core of the Iberian Pyrite Belt, which is a very one of the highest concentration of pyrite that we have on this planet. I mean, the outstanding properties of the river is the high concentration of iron. It can reach 20 grams per liter in the origin, and a similar concentration of sulfates. And the reason for that is because these are the products of digestion, of the metabolism of microbes that they chew pyrite, and they produce ferric iron and sulfuric acid. The combination of conventional microbial ecology, which is very badly needed in order to understand what microbes do, combined with molecular ecology tools that allows to quantify microbes, in this case microbes that they are sitting on pyrite. These green dots here are bacteria that they are very happy sitting on this type of pyrite. I mean, we can design, or we've been able to produce a model of how the Rio Tinto performs. It's a geomicrobiological model because, as you will see, it has biology and it has also geology. So we know that there is bacteria that can oxidize iron in aerobic conditions, and this can produce different types of minerals, which will be very important as a Mars analog. Also, we know bacteria that can entertain an iron cycle because they can reduce the iron. So the combination of these microbes can go forever, you know, if they have the means or the ability, the energy to do so. We know other types of microbes that they can do, they can perform in Rio Tinto. So these days we really know all the microbiology acting there, and what we can arrive to the conclusion that over there the two main cycles that they are in operation is the iron cycle and the sulfur cycle, which will not be a surprise because these guys live on pyrite. For the people that they don't have the chemistry in good shape, pyrite is iron sulfide. Also, the geo part of this geomicrobiology project, it has allows to do an analysis of the system because these bacteria, what they produce is a lot of minerals. The byproducts are minerals that you can analyze. Here you can see a collection of minerals. Here you have gerosite. Nobody cared about gerosite a few years ago. Now everybody wants to know what it is. I mean, you can see here goethite, spermanite, hematite. So the biology, the metabolism of these types of microbes are producing biosignatures. That, by the way, we were very happy to listen to Professor Squires when he reported that gerosite was found in Mars. I mean, this put really Rio Tinto on the spot. These type of minerals can be preserved on the rocks as a biosignatures, and also we are learning how to distinguish the ones that are produced biologically from the ones that are produced abiotically. What is very important to learn is just that biology doesn't do anything that chemistry doesn't allow to do. The only thing that biology does is to accelerate, to catalyze reactions. And normally, because biology performs at low pressure and temperature, the type of crystal minerals, the structure of the minerals are different from the ones that they are produced by pure geology. And something that has been mentioned this morning, the Marte project, has been going underground. I mean, all what I referred now before is just the description of the river by itself, which was the first thing to do. But obviously, after Rio Tinto became an interesting model for astrobiology, we decided that probably we need to know what happens on the subsurface, because it was very obvious for us that the quality of the river is not due to any chemical reaction that happens on the surface, but it's just the byproducts of a big underground reactor that is operating in the Iberian Pyrite Belt. I mean, still we are analyzing the data of the different wells that, in collaboration with NASA, we did going through the Iberian Periodic Belt. But what I can tell you today is that we found interesting activities that probably is the kind of activities that you might expect to find in a place like Mars. For instance, iron oxidizers, sulfur oxidizers, methanogens, and sulfur reducers. Mainly the two last ones, the methanogens and sulfur reducers, are very well known by microbiologists as strict anaerobic microbes. So these guys are operating very strict anaerobic conditions, and again, this puts Rio Tinto, or at least the subsurface of Rio Tinto, in a Mars context. If we now take all together the data that we have from different missions on Mars, especially in the Meridiani Planum, and the ones that we are collecting from Rio Tinto, as you will see, you have to pay attention. Obviously, I did this graph, on this table, and I select the things that are good to sell the product. But in any case, I think what is astonishing is to find out that in Rio Tinto, in the surface or underground, you find a lot of positive signals that you can, has been reported in Mars. Especially one that has been very interesting, and it's a nice byproduct of the Marte project, is to find methane production underground in these acidic conditions. So all these properties have promoted Rio Tinto as an interesting analog of Mars, and now what we are doing is to use Rio Tinto as a bench in order to test different methodologies, different technologies that are going to fly to Mars in the future. So there we have been testing this summer the XRD, which has been the ChemIn has been done very nice results on the geochemistry, and also the Center of Astrobiology is testing the SOLID. It's a life detecting system. Obviously, we know that in Rio Tinto there is life. What we need to know is if in Mars this life exists, and obviously this is something that we will need international collaboration to solve. Thank you very much.
Thank you very much, Ricardo. I think we have now stretched the links between Mars, the programs to search for life, and coming back to the Earth. And now it's the time to involve the audience into the plenary, and I would like to ask you whether you have questions or comments. Yeah, Bernard? Please say your name and then.
Yeah, Bernard Frank from the European Space Agency. For Ricardo, I have a question about the inhabitants of Rio Tinto. Do we have a description of the various populations? So what is really indigenous and what came from other origins which have adapted to those conditions?
Well, this is a difficult question to answer because I didn't have time with the 5 minutes that they give us. But the prokaryotes, the ones that they obtain the energy from minerals, it's very well understood. That is the kind of model that I put on the display. And we think that they are endogenous because they cannot live in another place that there is no pirate. But in addition to that, there is an incredible amount of eukaryotic diversity that they are able to live at high concentrations of heavy metals and at low pH. So this means, by all means, is adaptation because these are algae and fungi that are very recent in evolution. So, and they are very similar to things that we found in other places nearby. So this, I think, from an astrobiological point of view, it's telling us that life cannot only take advantage of some energy system, but also others can adapt once this has been transformed by the pioneers, in this case the chemolithotrophs.
More questions? Mr. Stritzel.
Roland Stritzel from Germany. My question is the following. Do you know there are investigations or experiments for creating life?
Sorry, experiments for creating life?
Yes.
Well, I think that there's, for more than 50 years, people have been trying to create life from bits and pieces, parts of the molecules that form life. so far, and this dating back to even before Stanley Miller's experiment in 1953, so far we haven't been able to do it. And I think the thinking these days is that, previously people were mixing specific molecules in specific situations together and hoping that a cell would appear. But these days people are going more towards putting just everything in the pot, a real primordial soup, and hoping that something will come out. I don't know if somebody else has something to offer.
There have been a couple of efforts that have been made in different directions. Self-replicating chemical systems, are things that people would like to be able to put together, but they wouldn't call those life even if they did. On the other hand, there's been an effort to synthesize a minimal genome and the minimum requirements for life associated with a very small, stripped-down parasitic-type cell. And there have been people looking at that. And there are a number of ethical considerations that people have been bringing forward on those. And there's some really good work done by, for example, Mildred Cho at Stanford on
that. Dick Klein?
Dick Klein. The question I wondered, you've indicated a lot of terrific programs that are underway, and I wondered what is the mechanism for coordinating the programs to get the most synergy out of the different things that you're doing?
I think this goes to the space agency representatives.
Mechanism is a interesting word. Certainly, the way we've been doing it for analog research has been scientist to scientist determining what would be the most useful thing, and basically out of the Center for Astrobiology and through the NASA Astrobiology Institute, scientists have basically got together, thought that, for instance, Rio Tinto is a good analog, and set it up that way. So that's on a person-to-person level or scientist-to-scientist level. Agency to agency, there isn't a specific mechanism. There's just different pathways of doing it. Part of it is the international IMIWG, which is International Mars Working Exploration Group. So once a year we get together and talk about things that we can do together, but also there's a lot of, let's say, bilateral discussions. And one of the things, because we both only had 5 minutes of time, and ExoMars is a good example of our intended collaborations for that mission. We also have scientists from both sides on missions that are ongoing on Mars right now. So it happens, but it is in some ways not a mechanism for doing it. And you may want to add to that.
Well, just to say that usually the process is initiated when you release a call for proposals. Both NASA and ESA make them open to the international scientific community. So that's a good way to gauge the interest from the other side of the ocean, so to say. And then once we have identified that, then we get together. But as Michael was saying, it's on a mission by mission basis. We don't have a permanent or an identified mechanism for dealing with this. We do it every time we invent it again.
May I follow on this question? We have now heard about how the cooperation works between ESA and NASA, but during this meeting we have learned that there are other spacefaring agencies who plan to go to Mars, either human missions or robotic missions, especially I think of Russia and China. Are there any attempts to build up a kind of coordinated consortium for these kind of missions?
Coordinated consortium. I mean, it is one of those things where negotiations go on, and it does happen on, agency to agency or, head of program to head of program kind of level. But there isn't a specific mechanism in which that's accomplished.
If there is not one burning question anymore, I have one question. There's one? Okay, yeah, please.
Steve Edberg, JPL. With respect to Rio Tinto, can you describe the variation in the various prokaryote populations as you move farther from the river and deeper into the ground, please?
Well, I mean, in the river I can describe it. If you are interested in details, I can give you a lot of details because we've been working there for 15 years and it's quite easy. The prokaryotic diversity is very low, so this helps to understand the system. Underground, that's much more complicated. I mean, subsurface microbiology is not easy, and we are still analyzing results. We can enrich cultures, and we also can quantify using fish, I mean, a hybridization system, but the density is so low. In some samples, we find only 2 cells per square centimeter. So this can give you an idea that the cell density that we are facing there, even in a system that is very active, is very low. So I think this gives us a hint that this will be a complicated business to do it on Mars.
So I will use the function as a chair and put forward one question which maybe some of you might have but might not dare to answer, and that is, assuming we find indigenous life, active indigenous life on Mars, are we well prepared how to handle it and how to, do we have the rights to go there? Is that an ethical question? Do the COSPA planetary protection guidelines are sufficient for this? I mean, we are now really going there and looking. Do we have enough knowledge and preparations how to deal with it? Maybe that goes to John.
Gee, Thor. No, Gerda, actually, people should come to the session on astrobiology that takes place shortly after this. Well, we'll answer that question. But effectively, the Coast Guard guidelines are focused currently on robotic missions. I think we're all in the same boat, but we've had some good interactions, particularly with ESA, looking at human-associated issues. One of the things that we've looked at is, in fact, making sure that any human-associated contamination wouldn't get into what we call Martian special regions, which are places where we think that liquid water could exist or where the high possibility of Martian life might be. But at this point in time, without a thorough robotic search for life, we really don't have the knowledge we need to safely put people on Mars. And I actually think that there are a number of people who would advocate a sample return as part of that preparation. But the National Research Council in the U.S. put out a document called Safe on Mars when they asked the question, do we need to know more about Mars before we send people there? And the answer was a resounding yes. Part of the answer has to do with whether or not there's life on Mars and whether or not you're likely to run into it. If you're an astronaut and you're going to be exposed to an unknown life form, you have the right to know under an ethical consideration called informed consent. So I think that even the flight surgeon would be a little leery of putting you into a situation where the planetary protection officer might tell you you can't come home. So we'll leave it at that.
Thank you.
Room 11.
I think with this, we hope we have increased your interest in the exploration of Mars, because we need all the public support for doing these missions. And I would like to thank the audience for participating. I would like to thank the panelists for giving us these interesting talks. And I would like to thank John for co-chairing it, and I hope that maybe in a few years we can give you real data about is there life on Mars, was there life on Mars, and so on. Thank you very much.